Good morning everyone. Today I am going to discuss about cytoskeleton, cilia flagella of cell motility and their organization and functions. Cytoskeleton. What is cytoskeleton? The cytoskeleton is a network of filaments and tubules that extend throughout the cell through cytoplasm, which is all of the material within a cell except for the nucleus. It is found in all cells, but the protein that they are made will differ between the organisms. The ability of the eukaryotic cells to adopt a variety of shapes and to carry out coordinated and directed movements depends on the cytoskeleton. The cytoskeleton extends throughout the cytoplasm and is a complex network of three types of protein filaments. They are microtubules, microfilaments, and intermediate filaments. The cytoskeleton is also can be referred to as cytomusculature. The cytoskeleton is apparently absent from bacteria. It may have been a significant factor in the evolution of the eukaryotic cell. Nikolai K. Kolzov proposed that the shape of the cells was determined by a network of tubules that he termed the cytoskeleton. He is the one who proposed the term cytoskeleton. The main proteins found in cytoskeleton are myosin, tropomyosin, actin. They are found in the microfilaments. Tubulin in the microtubules and keratins, vimentins, desmins in the intermediate filaments. Tubulin and actins are globular proteins, but the subunits like keratins, vimentins, desmin are fibrous proteins. First is microtubules. The microtubules were first observed in the axoplasm of a myelinated nerve fibers by Robertis and Frange in 1953. The microtubules are hollow cylindrical structure approximately 25 nanometer in diameter. They are seen in eukaryotic cells and plays crucial role in determination of cell shape and cell motility. A large number of additional cellular molecules called microtubule associated proteins MAP bind to microtubule and determines and control functions. Where they are found. The microtubules are found in all eukaryotic cells, either free in the cytoplasm or forming part of centrioles, cilia and flagella, except human erythrocytes, that is RBCs. The microtubules mainly occur in the sites like cilia and flagella, centrioles and basal bodies, axon and dendrite of the nerve cells, mitotic apparatus, elongated cells during spermatogenesis of certain insects, etc. Next is the structure and composition. A microtubule consists of a long and branched hollow tubules 24 to 25 nanometer in diameter, several micrometers long and with 6 nanometer thick wall having 13 subunits or protofilaments. The wall of the microtubule consists of 13 individual linear or spiraling filamentous structures about 5 nanometer in diameter, which in turn are composed of tubulin. Protofilaments have a center-to-center spacing of 4.5 nanometer. Application of negative staining technique has shown that microtubules have a lumen of 14 nanometer wide. The protofilament of microtubule is made up of protein called tubulin. Tubulin is an acidic protein with a molecular weight of 55,000. It occurs in two forms, that is alpha tubulin and beta tubulin, each containing 450 amino acids. These proteins have a distinct though closely related amino acid sequence and are thought to have evolved from a single ancestral protein. The tubulin in the form of dimers polymerizes into the microtubules. Thus, the heterodimers of tubulins assemble to form linear protofilaments with the beta tubulin of one dimer in all the 13 protofilaments are aligned parallelly with the same polarity. That means they are having charge, and the polar structures they with the polar structures having a plus or fast growing end and minus or slow growing end. The minus ends of cytoplasmic microtubules in the cells are bound tightly to microtubule organizing centers MTOCs, from which the assembly or polymerization starts. MTOCs also protect the minus ends of the microtubules from the disassembly. Generally, the plus ends of the microtubules terminate near cell margins and are protected from disassembly by the capping proteins. This is the structure of the microtubule, and this is the alpha tubulin and this is the beta tubulin. This is a diagram showing how tubulin pack together to form cylindrical wall. Next is the MAPs, microtubule associated protein. A number of proteins have been identified that associate with the surface of microtubules. These proteins are called microtubule associated proteins. The two major classes of MAPs are HMW proteins, that is high molecular weight proteins, and tau proteins. Both classes of proteins have two domains, one of which binds to the microtubules. Because this domain binds to several unpolymerized tubulin molecules simultaneously, these MAPs tend to speed up the nucleation step of tubulin polymerization in vitro. The other domain is believed to be involved in linking the microtubule to other cell components. Antibodies to HMW and tau proteins show that both proteins bind along the entire length of cytoplasmic microtubules. Next is the microtubule organizing centers, MTOCs. The microtubules are not found helter-skelter about the cell but are organized in specific patterns designed to carry out specific functions. Spontaneous nucleation, as seen in vitro, probably does not occur in vivo. Rather, initiation of assembly occurs at microtubule organizing centers. Thus, MTOCs are nucleating centers that serve as templates for the polymerization of tubulin. That means they are needed for the assembly of these alpha and beta tubulins. MTOCs exist in basal bodies. That is example in the chlamydomonas, in the centrioles of most animal cells, at the poles of mitotic spindles in dividing cells that do not have centrioles. That is most plant cells. Next is the function of cytoplasmic microtubules. First is the mechanical function. The shape of the cell and some cell processes or protuberances such as axon and dendrite of neurons, microvilli, etc. have been correlated to the orientation and distribution of microtubules. Second is the morphogenesis. During cell differentiation, the mechanical function of microtubules is used to determine the shape of the developing cells. The enormous elongation in the nucleus of the spermatid during spermatogenesis is accompanied by the production of any orderly array of microtubules that are wrapped around the nucleus in a double helical arrangement. Next is cellular polarity and motility. The determination of the intrinsic polarity of certain cells is also related to the microtubules. Directional gliding of cultured cells is found to depend on microtubules. Next is the contraction. Microtubules play a role in the contraction of the spindle and movement of chromosomes and centrioles, as well as in the ciliary and flagellar motion. Next is the microfilaments. They are thin, solid microfilaments of actin protein ranging between 5 to 7 nanometer in diameter and indeterminate length represent the active or motile part of the cytoskeleton. They appear to play a major role in cyclosis and amoeboid motion. The microfilaments are generally distributed in the cortical regions of the cells just beneath the plasma membrane. In contrast, intermediate filaments and microtubules are found in subcortical and deeper regions of the cells. They are found in the microvilli of the brush border of intestinal epithelium. Chemical composition of microfilaments. The actin is the main structural protein of microfilaments. The concentration of actin in non-muscle cells is surprisingly high. It may account up to 10% of total cell protein. It can be extracted and in vitro settings will undergo polymerization reaction from G-actin monomer state to F-actin. In fact, the globular, that is G-actin, and fibrillar, that is F-actin, transition is the basis of the classical Sol-Gel transition in the cytoplasm of moving cells. There are three types of actins, that is alpha, beta and gamma. The alpha form of the actin is found in fully mature muscle tissues. The function of microfilaments are: the microfilaments are found to be involved in movement associated with furrow formation in cell division, cytoplasmic streaming in plant cells, example Nutella and Cara, and cell migration during embryonic development. This is the structure of microfilament, and this is the F-actin, this is the G-actin. This is the monomer subunits, and they bind the polymerized actin microfilaments, that is the globular G-actin and F-actin together polymerize to form this actin microfilament. Next is the intermediate filaments. The intermediate filaments are tough and durable protein fibers in the cytoplasm of most higher eukaryotic cells. They are constructed like woven ropes. They are typically between 8 nanometers to 10 nanometers in diameter, which is intermediate between the thin and thick filaments in muscle cells where they were first described. Their diameter is also between microfilaments, that is actin filaments, and microtubules. The intermediate filaments are found resistant to colchicine and cytochalasin B and are sensitive to proteolysis. The various names have been also attached to the intermediate filaments that have a basis in the cell type in which they are observed. Thus, if these intermediate filaments are found in the epidermal cells, they are called tonofilaments. If they are found in the nerve cells, they are called as neurofilaments, and if they are found in neuroglial cells, they are called as glial filaments. The intermediate filaments have a tubular appearance. Each tubule appears to be made of 4 or 5 protofilaments arranged in parallel fashion. There are types of the intermediate filament proteins. That first one is the type 1 intermediate filament proteins. These are found primarily in the epithelial cells and include two subfamilies of keratin, also called tono, parakeratin and cytokeratin. Acidic keratin and neutral. They are acidic keratin and neutral or basic keratins are there. Keratin filaments are always heteropolymers formed from an equal number of subunits from each of these two keratin subfamilies. The keratins are most complex class of intermediate filament proteins with at least 19 distinct forms in human epithelia and 8 more in the keratins of hairs and nails. Mammalian cytokeratin are alpha fibrous proteins that are synthesized in cells of living layers of the epidermis and form the bulk of the dead layers of stratum corneum. Next one is the type 2 intermediate filament proteins. They include the following 4 types of polypeptides, that is vimentin, desmin, cinnamon, and glial fibrillary acidic protein or glial filaments. The vimentin is widely distributed in cells of mesenchymal origin, including fibroblast, blood vessels, and endothelial cells and White blood cells. The desmin is found in the both striated and smooth muscles. The glial filaments occur in some type of glial cells like astrocytes and some Schwann cells in the nervous system. The cinnamon is a protein present in the intermediate filament of muscles together with desmin and vimentin. The next is the type 3 intermediate filament proteins. These filament proteins assemble into neurofilaments, a major cytoskeletal element in nerve axons and dendrites, and consequently are called neurofilament proteins. In vertebrates, type 3 intermediate filaments consist of three distinct polypeptides, so called neurofilament triplets. Next is the type 4 intermediate filament proteins. They are the nuclear lamins which form highly organized two-dimensional sheets of filaments. These filaments rapidly disassemble and reassemble at specific stages of mitosis. Here is the table representing the types of the intermediate filaments. Here we can easily understand what is the composition and what is the location of this type 1 to 4 intermediate filaments. Next is the functions of intermediate filaments. The main function of most intermediate filament is to provide mechanical support to the cell and its nucleus. Intermediate filaments in epithelia form a transcellular network that seems designated to resist internal forces. The neurofilaments in the nerve cell axon probably resist stresses caused by the motion of the animal, which would otherwise break these long thin cylinders of cytoplasm. Desmin filaments provide mechanical support for the sarcomeres in the muscle cells, and vimentin filaments surround and probably support the large fat droplets in the